Hi, my name is Emily Landy, and I am a graduate student in Dr. Scott Cana's lab. And I'm excited to talk to you guys about how IL-18 and Perforin deficiency promote hyperinflammation in both an independent and synergistic manner. And so just to start us off, hyperinflammation is a life-threatening systemic inflammatory syndrome that occurs in multiple contexts, but is characterized by multiple clinical phenotypes, including recurrent fever, hepatosplenomegaly, hemophagocytosis, which is shown here, as well as inflammatory cytokine storm. And so our lab is specifically focused on how host susceptibility factors can lead to this disease in patients. And so today I'll be talking about two susceptibility factors. So under normal circumstances, when a cytotoxic T cell or an NK cell sees an infected cell, it uses perforin and granzymes to cytotoxically kill and eliminate that cell from the body. However, if you lack this capability of cytotoxic killing, you end up with a hyperinflammatory disease known as hemophagocytic lymphohistiocytosis, or HLH. And this is due to a dominant loss of function mutation and any number of genes that are related to cytotoxicity, so something like perforin. Now, another susceptibility factor to getting hyperinflammation is when you have a mutation that causes IL-18 to be released chronically and in excess. And this leads to extremely high levels of IL-18 that can even be found, that is found in the serum of patients. And this is clinically known as macrophage activation syndrome. And so while there's quite a bit known about how the lack of cytotoxicity can lead to hyperinflammation, how MAS patients end up with hyperinflammation is not as well understood. Therefore, the main focus of this first part is to understand the mechanisms behind the host susceptibility factors. The hypothesis here being that since HLH and MAS have similar clinical phenotypes, could they be sharing a similar mechanism of action? And so in order to do this, we are able to induce hyperinflammation in mice experimentally through infecting them with the murine virus LCMV. And here I'm showing you the data of LCMV infection in perforin knockout mice, shown here in red, which are representative of our HLH patients because they completely lack the cytotoxic perforin. And as well, I'm showing you here in blue, infection in IL-18 transgenic mice, which express super high levels of systemic IL-18, representing our RNAseq patients. And so when you infect both of these genotypes of mice with LCMV, you end up getting a hyperinflammatory phenotype that very well represents what we're seeing in patients. And so this includes hemophagocytosis, as well as high levels of pro-inflammatory cytokines, such as interferon gamma and IL-18, as well as very large spleens, so splenomegaly and anemia. However, one of the distinguishing factors between these two genotypes during the course of LCMV infection is that the perforin knockout mice actually cannot fully get rid of the virus in these mice. So these perforin knockout mice actually succumb to viremia. However, on the other hand, the IL-18 transgenic mice, which still have this very high hyperinflammatory phenotype, are clearing virus to the similar extent and of the same kinetics as a wild type mouse. And so this is suggesting that while the IL-18 transgenic mice are responding with a more autoinflammatory mechanism, the perforin deficiency mice are both have a immunodeficiency and autoinflammatory mechanism going on. And so this led us to wonder what could be driving the hyperinflammation in these mice that have such different viral clearance kinetics. And so I mentioned a couple slides ago that interferon gamma levels were very high in these mice. And so here we wanted to see if interferon gamma was driving this phenotype as a whole. And so when we depleted the interferon gamma during the course of LCMV infection in both perforin knockout and in IL-18 transgenic mice, shown here in the open circles, that we get a partial rescue phenotype. However, it's not completely rescued. So we took a step back and we're like, okay, what are the cells that could be propagating this phenotype here? And since we knew that interferon gamma was playing a rather large role, we opted to look at the cells that we knew produced interferon gamma. And so two of the main producers of interferon gamma are CD8 T cells and NK cells. And so whenever we depleted NK cells during the course of LCMV infection in both of these genotypes, we saw very little effect on the hyperinflammation. However, when we depleted the CD8 T cells, which is the data that I'm showing over here on the left, is that it largely reduces all of the hyperinflammatory phenotype that we saw. And so this is suggesting really that CD8 T cells are an important cell type for this hyperinflammatory phenotype that we're seeing. And so when we take a closer look via flow cytometry, we saw that the CD8 T cells of both perforin knockout and IL-18 transgenic mice are expressing more TNF and interferon gamma upon in vitro restimulation than in a wild type situation. And that they also have upregulated inhibitory receptors such as PD1 and CD39. However, the perforin knockout and the IL-18 transgenic CD8 T cells are not exactly the same. However, they both are still showing a hyperactivated CD8 T cell phenotype. However, here I'm showing you KLRG1, which represents short-term effector cells. And this is saying that the wild type and the IL-18 transgenic mice both have upregulated KLRG1s, meaning that they have short-term effector cells. However, the perforin knockout mice are not really showing this. And so we took a closer look and even deeper dive down into these CD8 T cells. And at the transcriptome level, we can see that the LCMV specific CD8 T cells from perforin knockout mice are wildly different from the transgenic mice as well as the wild type mice. And they are specifically showing a high increase in the levels of inhibitory receptor expression. And this is shown by PD1, CTLA4, LAG3, TIM3, and TIGIT, as well as TOX. And on the other hand, we have the IL-18 transgenic mice, which are showing much more of a terminal effector phenotype, which is a little similar to the wild type cells. And so this is shown by GM-CSF production, as well as TCF1, and again, KLRG1. And so since previous data has shown that the perforin knockout mice die due to the lack of antigen clearance, and the IL-18 transgenic mice were clearing just fine, we wanted to know if the IL-18 was directly acting on these T cells, and that was what was causing them to become hyperactivated. And so to do that, what we did is we crossed our IL-18 transgenic mice, so the mice that have super high IL-18, to a mouse line that had all of their T cells lacking the IL-18 receptor. So essentially, there's a whole bunch of IL-18 around, but all the T cells cannot see it and react to it. And so whenever we infected these mice with LCMV, they actually were nearly completely rescued from this hyperinflammatory phenotype. So in summary of just this first little bit, I've shown you that CD8 T cells are a key player in hyperinflammation, and that this could be due to direct IL-18 signaling on those T cells that's causing these cells to become hyperactivated. And so while we were in the research process of figuring out the mechanism behind this, which is still ongoing, there was some new and interesting data that came out. And this was that MAS patients who, again, have high levels of IL-18 all the time, also had a higher chance of also having heterozygous defects or partial defects in the genes that are associated with cytotoxicity, namely those same genes that we see that are completely defective in HLH patients. And so this made us think, what if cytotoxic defects in excess IL-18 are working synergistically to create hyperinflammation? And so to look at this, we crossed our perforin knockout mice with our IL-18 transgenic mice. And while mice that only had a partial deficiency in perforin but high IL-18, so these are the perforin heterozygosity, perforin het IL-18 transgenic mice shown in the pink, are surviving, we are seeing this spontaneously death phenotype in these perforin knockout transgenic mice, meaning that they completely lack perforin, but they have high IL-18. And then this is saying that like almost by day 50, we only have a quarter of these mice left. And so when we looked into why these mice were spontaneously dying, we saw that the perforin knockout mice, again, here in this purple color, perforin knockout IL-18 transgenic mice, were actually dying of hyperinflammation. And these mice were showing nearly all of the same symptoms of not only the patients that we see in clinic, as well as the IL-18 and perforin knockout mice when they were given LCMV. However, in this case, there is no infection going on. And it's also important to show that while the perforin het IL-18 transgenic mice, so these pink mice, which would more closely represent those MAS patients who have partial defects in cytotoxicity. Are not, again, not succumbing to spontaneous death, but are still showing this intermediate phenotype of hyperinflammation, which is very interesting. And so to better understand what was happening here, both in the perforin knockout IL-18 transgenic as well as a perforin heterozygosity counterpart, and on top of knowing the fact that CD8 T cells are a major player from the previous data, we wanted to look at what these CD8 T cells are doing. And so what's interesting is that this shows very clearly via flow cytometry that there's a population that is completely missing in these perforin knockout mice shown here, that becomes a very dominant population as you get to the perforin knockout IL-18 transgenic mice. And as it turns out, these looked very similar to our hyperactivated CD8 T cell population that we saw in our previous LCMV experiments. And so these, again, are CD8 T cells that are PD1, TIM3, CD39, and TIGIT positive, as well as producing a lot of interferon gamma. So these are like a vast amount of interferon gamma positive cells under, again, homeostatic conditions. And so what's interesting and exciting about this is that this increase in interferon gamma production gives an opportunity to block interferon gamma, and this actually partially rescues this phenotype as well. But what's really exciting is that interferon gamma blockade is in human trials and is ongoing and is super promising for HLH and MAS patients. And so when we looked at the transcriptional level of the profiles of the activated CD8 T cells from these mice, we see, and again, this is at homeostatic conditions, we see that the perforin knockout IL-18 transgenic mice are showing increased expression of inhibitory markers such as PD1, TIM3, and TOX, as well as they are the highest producers of interferon gamma and granzymes at baseline. And so this suggests that these CD8 T cells, especially in the perforin knockout IL-18 transgenic mice, are reminiscent of those that we're seeing in the LCMV hyperinflammation model that I was talking about earlier, except for these cells are spontaneously becoming hyperactivated. And so in the future, we're really hoping to further elucidate the pathways that are involved in creating and maintaining these hyperactivated CD8 populations. And then this way, further down the line, we can create therapies to better treat these hyperinflammatory syndrome patients based on the specifics of their individual disease. And so with that, I'll leave you with some take-home messages. So in comparison to a normal healthy response, patients with susceptibility factors, such as patients with high IL-18 or MAS patients, rely on the direct effect of IL-18 signaling on CD8 T cells, and that in patients with HLH or have a perforin deficiency in our mice, when they have impaired cytotoxic ability, they also lead to hyperinflammation. And on top of that, cytotoxic defects in conjunction with in conjunction with excess IL-18 could cause spontaneous hyperinflammatory phenotype. And that was shown in our perforin knockout IL-18 transgenic mice. And with that, I would like to thank our lab. Scott Canna is recruiting clinical and postdoc fellows. And I'll take any questions. Thanks.